this session we are going to study about development of monocot embryo. In our previous classes we already covered the development of dicot embryo. So in this particular session we just compare these two things. That means what is the basic changes of your development of your dicot embryo and how it is different from your monocot embryo, and simultaneously we just continue the development of monocot embryo step by step. So let's continue the development of monocot embryo. Development of monocot embryo. So we all know that after fertilization the zygote is formed and it is 2 mm nature. So our first step is the zygote. From the zygote all the embryo is formed. So here is your zygote, and which is diploid in nature, that means 2 in nature. And I also told you that whenever you draw this type of diagram, just mention the ends, that means your micropyrite and the chalazoid. Nextly, the division is takes place. In the same things happens in your diploid embryo also, that means your development of your dicot embryo. The monocot, in your monocot embryo also the same thing is happen, that means your mitotic division is takes place transversely, but the changes is in your dicot embryo 2 cells are formed, that means one is your basal cell, one is your terminal cell, but here 3 cells are going to form, and the formation of cells is like this. That means your first division is takes place, 2 cells are formed. Nextly, the second cell is again divided. That means among these two, a single cell is divided. Both the cells are not divided. A single cell is there, right? And 3 cells are formed. Here is your first cell, here is your second, here is your third cell. So we just name it. Here is your chalazoid, here is your micropyrite, here is your basal cell. That means the cell which is towards your micropyrite is the basal cell. So here is your basal cell. Here is your terminal cell. That means the cell which is towards your chalazoid is your terminal cell. And the middle one is the middle cell. So a single change is takes place over here. That means from your monocot to your dicot. In dicot 2 cells are formed. In monocot the third cell is formed. That means your middle cell is formed. Next, the major change is that the terminal again forms the cotyledon. That means from the terminal cotyledon is formed. And there is no changes takes place in a basal cell. A single change is takes place, that means in size. That means in size the basal cell is after taking some nourishment it looks a little bit bigger. That is the change, but there is no major change takes place. The major change is takes place in your middle cell, which plays a vital role in your monocot embryo. But in dicot embryo, basal cell plays a vital role. I think you remember these things. That means from the basal cell the suspensor cells are formed. That means 7 to 8 cells are formed, and then next the posterior cells are formed, and a number of things. That means a number of things that changes in your basal cell. But here simply the basal cell is looks a little bit bigger. That's it. So here the middle cell plays a vital role. That means most of the changes that takes place. That means most of the changes takes place in your monocot embryo is changes from your middle cell, and the changes are the formation of a plumule first. The plumule is changes from here. That means forms from the middle cell, and the radical is also formed. Radical is also formed. That means from your middle cell. Nextly, the epicotyle is formed. The hypocotyle is formed. And lastly the suspensor is also formed. So these are all these things which are changed from the middle cell. What is the things? The plenial cell is formed, the radical cell is formed, the epicotyl is formed, the hypocotyl is formed, and the suspensor cell is also formed from the middle cell. So it plays a vital role in your monocot embryo. I think it is clear to you. Nextly, we just change the color to a better understand to your concept. So here is your basal cell, and I told you that the basal cell is as it is, but its size is little bit bigger. So here is your basal cell. Next, what happens? The middle cell is formed, and as I told you that middle cell plays a vital role. So a number of changes are takes place in your middle cell. Here is your middle cell, and it divides in all planes to form a mass of cells in first place. That means from the mass of cells, a number of things takes place. That means your plenial is going to develop from here, the radical is also developed from here, the epicotyl is also formed, the hypocotyl is also formed. So all these things are formed from the middle cell. Here is your basal cell, here is your middle cell. Next, the terminal cell. Terminal cell simply form your cotyledon. A single thing is formed over here. So here is your cotyledon. So this type of things are changes from the basal cell to as it is, the middle cell to all these things, and the terminal cell to cotyledon. So this type of structures somehow formed. I also told you that all these things are not look like that. For your better understand, we just do these types of things. So here is the changes takes place. Nextly, we just level it like that. From the here is the cotyledon part. All these things, including your suspensor cells, are formed from the middle cell, and the basal cell is as it is. So this structure is formed. Nextly, we just arrange the structures with the plenial, radical, epicotyl, hypocotyl, and important. Whenever I told you that the plenial and radical, these types of labelings are not mentioned in your monocot embryo. There is a slight change, but I told you the plenial and radical, the reason behind it, that in our previous classes in dicot embryo, we also know about the plenial and radical. And here the plenial and radical, these names are not mentioned. If the little bit change, that means the plenial is forms your collectin and the radical is forms your polar region. Why these changes are takes place? I told you a couple of stages later. So here is the next structure. The basal cell is formed as it is. After the divisions, the basal cell is as it is formed. I told you that there is no changes that takes place in the basal cell. That means a little bit bigger cell is formed as such. Nextly, the middle cell. That means the major changes which takes place here. And after changing of all these structures, it somehow look like this. So here is the middle cell, which is divided to form this structure, and nextly the cotyledon part. I again repeat these things. The middle cell forms your plenial first, next the radical. So here is the plenial part, and the plenial part is covered with. That means it is your middle cell. That means this total structure is your middle cell, which forms over here. So it is your middle cell. Nextly, the plenial is formed. So, but the plenial is covered with a number of cave-like structure, that means your seeds, which is found the structure which is known as your collector. It is the plumule, but because of these seeds, it is known as your collector. It is named as your collector. So here is your collector. Nextly, in your lower part, I told you the radical, but it is otherwise known as your coleoregia because of the formation of your cap-like structure, that means cap-like seeds, and because of these seeds, it is known as your coleoregia. This structure is your coleoregia. So, firstly coleoptile forms, nextly coleoregia is formed. So, the lower part of the coleoptile, that means lower part of the plumule in your dicot embryo, you also know that it is always known as your epicotyle. So, this place is known as your epicotyle, and nextly the part of your cotyledon. The cotyledon is attached to your cotyledonal node and somehow like this. Here is your cotyledon, and it is your singly peppery thin cotyledon. That means you mostly found this type of cotyledon in your grass, and in grass it is known as a scutellum. So it is your cotyledon, and in grass it is known as a scutellum. I also go to over here. Here is your scutellum. So cotyledon is formed, which is commonly known as a scutellum, and the point of the attachment of the cotyledon is already known as your cotyledonal node. So this place is your cotyledonal node. This place. And the lower part of the cotyledon is already known as your hypocotyle. So this part is your. So this type of basic changes are takes place. Nextly, a single change takes place here. That means the attachment of your suspensor cell. I told you that suspensor, I just change the color. The suspensor is also attached to the middle cell. So here the suspensor cell. There is not enough work of the suspensor cell, and it is little bit small in nature. So this type of changes takes place in your embryo when it is developed. That means when the embryo is developed. So this type of changes are takes place. So the plumule is changes to your colectile. The radical is changes to your coleoregia. The names are changed because it covers with seed. So the name which is covered, that means your radical is already known as your coleoregia. The colectile, that means the plumule which is covered with the curved seeds, which is already known as your colectile. Next, all the things are as it is. That means the place over here, that means the between the cotyledon and the colectile is already known as your epicotyle. That means from your cotyledonal node, and below the cotyledonal node, the all places are known as your hypocotyle and the coleoregia. That means your radical part. In your embryo axis, I also told you all these things. But the basic change is your colectile and the coleoregia, and the reason behind it, the seed which is covered. And a singly peppery cotyledon is also placed over here with attachment of your cotyledonal node, which is already known as a scutellum, and the scutellum name is because of the grass. In grass mostly these types of names are used, and it is singly thin peppery cotyledon, single cotyledon, as we are studying here, monocotyledon. So these type of changes are takes place. Next, what happens? This structure is not look like your monocotyledon embryo, so I just place these things with a seed. I just already draw an example of your maize seed, so we just place it little bit couple of stages later. So first draw the diagram first. So what happens? Nextly, the total structure, that means your basal cell and the synovium. So this is your basal cell. And this is your suspensor cell, sorry, synovium. This is your suspensor cell. These two cells, that means your basal cell and the suspensor cell, both are degenerate after the formation of all these things. That means after the couple of stages when the monocot embryo is going to form, nextly these two things are degenerate. But in your diploid embryo, suspensor plays a vital role for pushing the embryo for the nourishment, but here it is degenerate because there is not enough food material in your cotyledon. The reason behind it, as it is your thin papery and all these things I also told you over the example. Just wait and watch. So here all these things happen. If we degenerate these things, this total structure is somehow look like this. So here is your collected, that means it is covered with sheath. And here is your coleoids. Here is your coleoids. Nextly, that means your pleural part and radical part. And here the cotyledon which is attached. Here is the cotyledon. So, in lot of books, you look a number of changes, a number of diagrammatic representation also. So, just look this diagram, how this is placed in your monocot embryo. So, the two things, that means your suspensor cell and the basal cell, both are degenerated. Nextly, we just place the total structure, that means total embryo, to a seed of a monocot. We just studying about your monocot embryo, so we just take an example of the monocot seed. In monocot embryo, in monocot seed, we found the example of your maize seed, and the outer layer of your maize seed is so the outer layer, that means this thin layer is otherwise known as your alluron layer, that means the outer layer of maize seed. And after the alluron layer, you look that there are two compartments are formed in your maize seed. And the bigger one is for the starchy endosperm. That means it contains a number of nutrient material, is otherwise known as starchy endosperm. And the other one is your alluron. In morphology, you already know a lot of things about the maize seeds and a number of things for how the endosperm is placed and all these things. I just place the endosperm first here. So we just place the endosperm in your little compartment. So we just place the endosperm first here. Here is the coleoids, that means the cat-like structures, and here is your coleoids. Coleoid, and here is your, just draw it here. Here is your coleoid, here is your coleoid. Nextly, the attachment of your cotyledon, and here the cotyledon is attached. Here the thin papery cotyledon. So a question arises in mind that basically cotyledon resolve the nutrient medium, and the nutrient medium is for the embryo. That means for the cotyledon is the main part where the nutrient medium is formed. That means you may take your example of your pea seed. So why this thin cotyledon is that means the thin structure is, why the thin structure, that means the scutellum is already known as your cotyledon. The reason behind is that whenever we talk about the starchy endosperm, you may take the example of your maize seed itself. It contains a number of nutrient medium. So the endosperm only passes through the cotyledon. That means whenever the changes that takes place in your endosperm, the nutrient medium mainly transfer to the embryo. That means this is your embryo is formed and this is the endosperm, and the endosperm is transferred by the cotyledon to the your embryo. So the cotyledon plays the vital role here. So the cotyledon is placed over here, and the reason behind it, that means through it the nutrient medium is only passes. That means through it the nutrient medium passes to the polypeptide and polymerase, all the structures. That means for your embryo. So from the endosperm to the embryo, there is a single cotyledon is formed, which is thin and papery in nature. But through this thin and papery cotyledon, all the nutrient mediums are passes. So this is all about the changes which are takes from your zygote to a monocot embryo. I think all these things are crystal clear to all of you, and this video session is helpful to you. So in our next class, we are going to study about the end product of your chapter, that means your sexual reproduction in flowering plant. As you studying about all these things, the end product is your fruit and seed. In basically all these things we already know in your morphology chapter, but a little bit things are included in your sexual reproduction in flowering plants, because it is the ultimate condition. After the embryo and endosperms, all these things are formed, the flower is going